Very good morning to all the participants and respected resource person. Today's our eminent personality, senior most botany fraternity of our entire Telangana, Associate Professor Dr. THIRUNAHARI UGANDHAR sir, working in Kakatiya Government College, Autonomous Hanmakonda, Warangal district. Sir previously worked in various colleges, reputed institutions like SRR Government Arts and Science College, Autonomous Karimnagar, and Government Degree College, Mehbubabad, and Jagityal. So, sir has huge experience in the field of plant sciences, especially molecular biology, genetics, biotechnology, plant tissue culture. So, sir is going to explain in the today's class, that is the important cell organelles of our plant cell, that is nucleus and its components, and other most important aspects regarding of the CPGET 2025, and also the all competitive exams related to junior college lecturers, degree college lecturers, Gurukula degree college lecturers, Gurukula junior college lecturers, PGT, అదేవిధంగా TGT, వీటికి సంబంధించినటువంటి అన్నిటికీ ఉపయోగంగా ఉండే విధంగా, దాంతో పాటుగా అన్ని పోటీ పరీక్షలకి ఉపయోగపడే విధంగా మనకు చాలా ఉపయుక్తమైనటువంటి పీపీటీతో సారు సిద్ధమవుతున్నారు. వాస్తవంగా సార్ మన కోసం మంచి పీపీటీని స్పెషల్గా మన అందరి విద్యార్థుల కోసం, రేపటి యంగ్ బడ్డింగ్ బాటనిస్ట్ కోసం సార్ ఇంత మంచి పీపీటీస్ని మనకు ప్రజెంట్ చేస్తున్నారు. Sir, please welcome and start your session, sir. Please welcome. Sir, am I audible?
Okay, start there. So now, nucleus and its components. So what are the meaning of the nucleus, and how many number of the nucleus in cells? What type of the nucleus? What are the function of the nucleus? How many components? How many parts of the nucleus? Today we are discussing that the nucleus is a major important role in the cell, in the plant cell or animal cell. So this nucleus is a majorly which is located in the center part of the cell, in the protoplast or peripheral part. It can have only single nucleus or more than one nuclei, which is also the nucleus is which is performing all major functions in cell. It can be majorly performing all metabolic activities. Also maintains the cell division, cell differentiation, cell elongation, and also carries inheritance characters from generation to generation. Also, it can be important role into the mutations and variations. Every part of the living is a basic structure, functional, developmental unit, also called a molecular unit, is also called a cell. So, which is today we are discussing this the history of the nucleus, then occurrence and position, morphology, ultrastructures. Then ultrastructure means it can studies under the electron microscope how many parts of the nucleus are there, what are the important role, what are the important component, and then made of the components in the cell also. Then last one is also functions. Then after functions, then multiple choice questions are filling the blanks for preparation of the PG and JL coachings. So this is also what is the means of the history. The history is also basic. Then first discovered the name by Robert Brown, scientist, who was a famous scientist. That person is a total and the structure of the cell, then basic fundamental unit of the cell. Also the center part of the cell which can forming the nucleus. That is also first reporting. First discovered the name by the Robert Brown. The term nucleus Bowman, the second person, The Bowman is also in 1840. Then term chromatin, W. Fleming, 1817, and third person. Then fourth one, term cytoplasm and nucleoplasm, Strasburger, 1882. The cell which have differentiation into the cytoplasm and nucleoplasm. These nucleoplasm is which can consisting of the nucleoplasm nucleides. These nucleoplasm consisting of the RNA and DNA that is located in the center part of the nucleus, which are present in the chromatin bodies, which can float on the nucleoplasm of nucleus. This is also major studies by these four scientists. First, then Robert Brown, scientist, 1833, Bowman, 1840, Fleming, 1879, also got Strasburg, 1882. Then first scientist is also 1833, this are 1882. This is also famous sign. Then what are the occurrences and position? Occurrences means what is the located, which part of the located in the nucleus, means the nucleus are present in the every cell of the living organism of both prokaryotic and eukaryotic. In prokaryotic, the nucleus are not well developed. In eukaryotic, well developed. That is also difference. So in the last, we have also studies in differences between the prokaryotes and eukaryotes in separate. This is also important to the how to develop of the first organisms of the and evolutionary pre-existing cells also got a developing arch. And in the primary and also advanced characters of the organisms, how to development that can base of the important structure of the nucleus. This structure is major important role for the group of the plants which can classify the prokaryotes and eukaryotes. That is also important. You have that the nucleus layers, the nucleus parts, it is a major important. It can occur, it can found in all eukaryotic cells in plants and animals. In a prokaryotic, the nucleus is a not true nucleus, which is also called nucleide. That is also the nucleide only having a single nucleus, single DNA thread. That is, then exceptions, then mature sieve tubes and erythrocytes. The mature sieve tubes, which are present in the phloem of plants, then erythrocytes or BC, which are absent in animals. In prokaryotes, bacteria lack true nucleus. The prokaryotic and eukaryotic both have nucleus, but here prokaryotic, which are lack of the true nucleus, the eukaryotes are present of the well-developed nucleus. That is position, usually at a center, but change time to time according to the metabolic states. The metabolic states means during the cell division, prophase, metaphase, anaphase, telophase, the stages of the cell divisions based to the nucleus are which can turn into the protoplast. Sometimes are peripherally, sometimes are center. That is also then staining process. How to absorption of the nucleus. The nucleus are which can understand or observe and structurally reported with the help of the staining. That staining process are identified with under the electron microscopes. First you stain that part is observe under the microscope, that parts are clearly seen. So the fluorescent dyes with the aromatic amino or guanidine groups, such as propydium iodide, PI, ethidium bromide, EP, diaminophenyl alidinol, DAPI, acrylamine orange, AO, and oakist dyes interact with the nucleotides into the emit fluorescence. These parts which can add to the cell, also called a group of the cells, which can be stained. Stained means it can coloring, that can in the saffron type of the models, the saffron, which have in the transfer sections, the saffron parts are and light brune color is also changes in the same way. The staining process, which is important for identification of the nucleus and also called the properties, studies of the properties of the nucleus. This is also got a morphology, number. What is the means of the number? The number of the nucleus, how many number of the nuclei present in the cell, that is important. So, examples you say that is a mononucleated cells. And a majority of the species of the plants which have only single nucleus, but in sometimes two nuclei, sometimes three nuclei, sometimes more than three. That is called polynucleated cells. Then the binucleated cells, example paramecium. The paramecium which have macronuclei, two types. Then hepatocytes, also called chondrocytes, which have two types of the cells, small nuclei and big nuclei, two types. But here is only a small nuclei, micro, then big nuclei, macro. That is also differentiation. Then, so trinucleated cells, you know that the three nucleus, primary endosperm nuclei, which are also called then the two nuclei of the male, and a double two nuclei of the center part is fused with the one male nuclei to form the trinucleated primary endosperm nuclei. That is also one type. Nuclei number. Then polynucleated cells 3 to 100. Animals, so syncytial cells means also called osteoblast. Stated muscles fibers. In the plants, sinusoids. Sinusoids means multinucleated cells which have algae. In algae loop, then watch area, also called a fungi, rhizopus, mucor, which is also important in algae loop watch area. Then xanthophysi members, which have more than one nuclei, multinucleated cell, also called a sinusoidal cell. In a fungi, so rhizopus, mucor, which are also myconiana glass. That is also called a, so 8 nuclei are present in myconiana. That is important. The shape, what are the shape? The shape of the cells is also different. Majorly also called spherical, rectangular, triangulars, and also called heptagonal-like structures. The shapes are spheroid, cuboid, isodiametric, and ellipsoid. Example, squamous epithelium, discoidal shape, leukocytes, also called irregular cells. The shapes means then angles of the shapes are different based on the angles. The cells, which is also trigonal cells, hectagonal cells also dividing, then irregular in shapes. This is also one character. Then size. Generally, it can occupy the 10% of the cell volume, the total percentage of the cell, which is also 10%. Remaining 90% is other cell organelles are there. Means majority of the size, majority of the percentage is more. That is 10% of the part, which is called a nucleus. The size varies from 3 millimicrons to 25 millimicrons, depending on cell types. Then different type of the cell types means then animals, also plants, also bacteria and virus. Algae, fungi, bryophate, that the parts of the plants, majorly angiospermic plants, which have 3 millimicron to 20 millimicrons in size. The size of the nucleus is directly proportional to the cytoplasm. How much percentage of the cytoplasm is occupancy? The cytoplasm inside only single nucleus are present. That can basis the proportional to the cytoplasm. It is also heritage gives a formula for the determination of the size. Heritage sounds so the formula. So this formula is also got a VN, means NP BC minus VN, where NP means nucleoplasmic ratio. NP, N means nucleoplasm, P means plasm, BC means volume of cell, VN means volume of nucleus. Size is related with the ploidy of labor. these equations Vn, and then Np is equal to Vc by, and Np is equal to Vn by Vc minus Vn. This is also equation to the determination of the size of the nucleus. So example, applied cells have only small size nucleus, then diploid and polyploid cells, and vice versa. And that means that it also applied, and which part of the cells have applied in plants, pollen grains. Then also got after microsporogenesis, after reduction divisions, and one microspore mother cell undergoes meiotically to form 4 microspores are present. Each microspores are which can formation of the pollen grains. Then spore tetrads, that cell have only single nucleus. That is called a XY chromosomes are present in the ovary. Also got a microspore mother cell undergoes meiotically to form a 4 haploid microspore mother cells. These microspore mother cell 3 is also degenerated, only single is also replicated in the macrosporogenesis. So these macrosporogenesis are which have only uninucleated, haploid nucleus are present. Then the size depends on cell volume, amount of DNA and protein metabolic phase of the cells. So how much volume, how much amount of the DNA which can be based to the cytoplasm and proportional, and the proportion, the size of the nucleus proportion to the cytoplasm. The cytoplasm percentage of more, then size of the nucleus is also called more. That can depends, which can be related to the size. Then it is also got a structure of the nucleus. It is also got a electron micrography of nucleus. So which are different. What is the means of the structure in the based on the nucleus, which have only 4 parts are there. Outer layer means nuclear membrane. So these outer nuclear membrane, which can be, and then also called distinct small holes are there. So these holes is called nucleopores. Nucleopore. Then also inside of the nucleopore, that is also called nucleoplasm. The nucleopore outside a small and a thread-like structure, which is also called endoplasm reticulum. On the endoplasm reticulum, a ribosomes are present. These ribosomes are been present on the endoplasm reticulum, that is also called a rough endoplasm reticulum, RER. Then without ribosome, there is smooth endoplasm reticulum, that is important of the outside. So then the nuclear membranes are not entirely closed. When some places which are not closes, it is also got open. That open pore is also nuclear pores. Then inside of the chromosomes, inside of the nucleoplasm, which can consisting of the chromatin bodies and nucleolus, chromatin bodies, nucleoplasm, and nuclear membrane, majorly 4 parts. Then the nuclear membrane, which consists of the nuclear pores, that is also intranuclear chromatin, then nuclear envelope, then inner membrane or outer membrane. 2 members are there. Each the nucleus, which is a semi-permeable membranes, which can similar to the plasma membrane activity. Then plasma membrane activity is also, it is a major important characters of the cell or not. Then so which is also the nucleus. The nucleus is the next part. That is the alta structure of the nuclei. This alta structure of the nuclei, which is a character features of the nuclear envelope, enclosed DNA, formed form. Then second one, spherical inner nuclear membrane, that is 5 to 10 nanometer thick. It is also semi-permeable. It contains a species of proteins act as binding sites, which is also nuclear lamina. Outer nuclear membrane, which is a contains a with endoplasm reticulum, studied with ribosomes and proteins. So perinuclear space 10 to 15 nanometer diameter. It contains a fibrous lipids and also called a droplets or crystallines or deposits. Then also got a nuclear lamina. So what is the means of the nuclear lamina? The nuclear envelope is also outer nuclear membranes, inner nuclear membranes. The inside is also got nuclear lamina. It protein meshwork 50 to 80 nanometer thick lines inside surface of the inner nuclear membrane. It can play a crucial role in the assembly of the interface of the nucleus. So this is also just one which can clearly seen in the nuclear membrane and how many parts of the nucleus which is also there. So this is also the nuclear envelope. Only this part is a nuclear number. It outside is the observation of attached to the ribosomes here, particularly in the endoplasm reticulum lumen, which is also HP1, LP2. This is all is also got a tubules or microtubules are there. So these are microfilaments are there. This is microfilament. This is also nuclear envelope, means outer layer or covered by layer of the nucleus. That is called a nuclear envelope, this total part there. Then also called a nuclear pores. In the nuclear membrane, some places are open places which have nuclear pores, which are the circular diameter 10 nanometer to 100 nanometer in size, small, which can the nuclear pore complex means molecular weight is 50 to 100 million daltons, have octagonal symmetry. It can contain say 2 rings or large particles that forms a central plug and radial spores. This is also called a protein. And in the nuclear membranes, nuclear pore complex is also made by the protein structures. So these proteins are integral membrane protein, a glycoproteins of 120 kilodaltons. Then transport protein 63 kilodaltons. This too is also got a protein qualities. Then the second point, pore density. Density means the open space, open pore size, also got a space, how much space to open the nuclear membrane to cytoplasm. That is also pore density. Then correlated with a transcriptional activity of cell, RB and CS, lymphocytes, red blood corpuscles, and lymphocytes, 3 pores. Then also proliferation cells, livers, 15 to 20 pores or millimicrons. The pore is which can responsible to the then transport of the nucleoplasm to cytoplasm relations are there. So this is also important of the nuclear pore complex. Yeah, nuclear pore complex that outer part is also cytoplasm here. This is also called as cytoplasm. Particularly in the nucleus. You see, nucleus. The nucleus outside, then inside nucleoplasm. Between 2 layers. Outer nuclear membrane, inner nuclear membrane, between perinucleated space. That is also cytoplasmic filaments, then cytoplasmic rings, outer nuclear membrane. Here is also inner nuclear membrane. Between this is also central transport, nuclear ring, nuclear baskets, and nucleus. This is also nuclear pore. This nuclear pore is the nuclear pore complex is a nuclear pore, central transport, nuclear ring, nuclear baskets. This total is also got a nuclear pore complex. This nuclear pore complex is majorly important character features which can transport through the nuclear pores. Then in cells, synthesis of DNA, it needs to the important import about 10 to the 6 histones molecules from cytoplasm every 3 minutes for a package of DNA. Every 3 minutes, the DNA is a packages. Means it can be spirally arranged during the cell division, the metaphase, only clearly absorption of the chromosomes. The before prophase, then also called a telophase, anaphase. The chromosomes are thick and small in sizes that can movement That is also got every 3 minutes it changes of the nuclear pores. Then histones, nucleus, and cytoplasm every 3 minutes for packages of DNA. DNA packages and nuclear pore structure is also, it can be changes. mRNA complex with special protein forming ribonucleoproteins particles are actively exported through the nucleus. Ribonucleoproteins, which is important for the activity of export, also import, both are there. Export means it can be the inside of the nucleoplasm, which can be transferred to the through the nuclear pore to cytoplasm. Then cytoplasm to nucleopore into the nucleoplasm. In both, also called export. That is, it movement of the granular material presumed to be ribosomal subunit through the nuclear pores, which is a important of the nuclear pore complex. This is also transport of the protein through nuclear pore complex. This nuclear pore complex. Then here is also nuclear pore in the several type of the parts of nuclear membrane here there. So it is also imported protein. Imported protein. This is also got exported proteins. This is total structure is also got transport of the proteins through nuclear pore complex. Then is nuclear pore complex, which is important for the only mainly it can be the concentration of the ions of cytoplasm and nucleoplasm to clearly the same transport of that is one character. Here is also nucleoplasm. Then, so this nucleoplasm is majorly the transparent semi-solid granule, slightly acidophilic nature. Acids, sodium hydroxide. Then acids and bases are there. Also called the nuclear sap. Nucleoplasm also got a karyolymph. It can composed of the nucleoplasm composed of the two parts, nucleic acids. These nucleic acids are, which is a major important for the deoxyribonucleic acid, ribonucleic acids, RNA, DNA, both are major molecules, which can study the biomolecules. The biomolecules are present in the nucleus. In the nucleus, nucleoplasm. So this nucleoplasm is a only thread-like structures is called deoxyribonucleic acids, ribonucleic acids. Then inside of the nucleoplasm, it is another type of the components is called proteins. These proteins complexes, proteins are categorized into the proteins are which can categorize into the several types. The basic proteins. It takes a basic stains. So the name example nucleoprotein and protamines, molecular weight. Then DNA. Then also got a nucleohistones, molecular weight 10 to 18 kilo. Then also got a second non-histones. And the histoproteins, non-histoproteins. Then histoproteins, which is also called the nucleoprotamine, then nucleohistones. Then second one, non-histones are acidic proteins. Example, phosphoproteins. And the histoprotein, non-histoproteins, these are also classified the two types by the protein components in the nucleoplasm. The nucleoplasm is a majorly consists of the two parts. First one, nucleic acids. Second one is also called proteins. The protein also further divides into the histoproteins and non-histoproteins. This is also then third bite enzymes. Only majorly 5 type of the components which can dissolve into the nucleoplasm. First nucleoplasm is present in the nucleic acids. Second one is also proteins. Third one enzymes. Fourth one lipids. Fifth one minerals. These are all type of the components is called the nuclear components. These nuclear components which performing all metabolic activities genetically, non-genetically, morphologically, also got a physiologically changes qualities which are there That is also biomolecules, DNA, RNA, major role to the inheritance character. Then physiological characters, so transfer to the parent and to the daughter generation. The second one, that character is transferring with the help of the proteins, also got enzymes, also lipids, also minerals. These are all is important role to the living part, and also inheritance characters are improvement from primitive organism to advanced type of the organisms. This is there. So then enzymes. How many type of the enzymes are there? It can contain the enzymes presence for DNA ligase. RNA ligase means DNA synthesis. RNA synthesis means formation of the DNA, which is also semi-criminal, and in the semi means half of the chromosome parts, which is a conservative process. Semi-conservative process, the DNA is multiplied, single DNA to 2 DNA. That can process of the enzymes is related, secreted in the nucleoplasm. That is called a DNA ligase, or RNA ligase, or DNA synthesis. Or then example in the polymerase. This is a polymerase, nature of polymerase enzymes. In the PCR, TAC polymerase enzymes, which is a within a short period, large number of DNAs are multiplied. That is called TAG DNA polymerase. It is also naturally polymerase enzymes, nicotine, adenine, dinucleotide synthase, adenosine, so deaminase, guanase, aldolase. These are major enzymes which can performing the DNA activity and DNA multiplications and DNA inheritance characters are improvement. These are all is there. Also contains a cofactors and enzymes with like a ATP and acetyl coenzymes. These cofactors which can important for increasing the activity in the enzymes, do not participate the reaction, which only increasing rate of the reactions, rate of the enzymes activity. That is called a cofactor, ATP or acetyl coenzyme. Then fourth one is also called lipids. It is a small lipid contain a Sternberg 1837 and Dawes 1955 reported the lipids. Then fifth one, last components of the nucleoplasm is called minerals. These are minerals are important. First unit in the plants, carbon, hydrogen, oxygen, nitrogen, phosphate, calcium, potassium, magnesium. These are all types is also important. Which are also fifth major important element is also got a phosphate. You know that is also got a minerals, contains several inorganic components such as potassium, phosphate, potassium, sodium, calcium, magnesium, which are a major present in the nucleoplasm. Additionally, then carbon, hydrogen, oxygen, which are easily available in the plants in the form of a carbon dioxide. Then carbon dioxide, carbon, oxygen, both are there. But hydrogen oxide, water, hydrogen oxygens are there. Means 5 important elements are easily available in every part of the plant. But without the observation of the minerals from the soil. But here is a phosphate, potassium, sodium, calcium, magnesium, which are definitely transferred from soil to plants. Soil to plants. This is a important of the nucleoplasm reactions are there. Then also called a chromatin fibers. The third important part is called chromatin. Means what is the means of the chromatin is a fundamental part which can formation of the chromosomes. During the cell division, the chromatin bodies are elongated to become a chromosomes. These chromosomes coloring threads which carries a inheritance characters from generation to generation. It can also present in the DNA, also in on the DNA, which are genes. This is also characters. Then also the chromatin fibers thread-like coiled and much elongated in structures. During the cell division process, it takes a basic stain like a basic features observed only during interphase. Interphase means between the two cell divisions, prophase, metaphase, anaphase, telophase. Then also got interphase. Then interphase also one of that. Then prophase, metaphase, anaphase. The prophase between prophase before, then G1, G2, yes, synthesis. So that is growth 1, growth 2, and synthesis. This is also interface is also between the two different cell divisions between. Then first one is the chromatin bodies, which can classify the two types, heterochromatin, euchromatin. Then what is it means? Hetero means hetero different, dark in color. So this darkly stained condensed region supposed to be metabolic activity, genetically inactive. That is there. Means dark color of chromatin which have present, that is called genetically inactive because it contains a small amount of DNA and large amount of RNA. That cannot, that only euchromatin lightly stained, diffused, then regions genetically active. Then it is a large amount of DNA are present. And it means then dark color of the chromatins does not have DNA more percentage, then less percentage, then also more percentage of the DNA, which can important role to the transfer of the genetic characters from generation to generation. That is called a euchromatin. The euchromatin means nija, and in the two, then hetero, the also got different, more dark in color. That is also the basically euchromatin active and heterochromatin inactive. Means the in heterochromatin less percentage of DNA, more percentage of RNA. Then euchromatin is also got a more percentage of DNA, less percentage, less amount of RNA. It is also two different. It is also important character of the this is also chromatin bodies. What are the structure of the chromatin bodies are here. So this is also the spaghetti model for organization of the chromatin in the interface cell nucleus. It is also dark color and also thread-like structures which are responsible to the chromosome formation. Here is the last one. Each chromosomes, that is called nucleosomes. These nucleosomes outside, it can formation of the DNA structures. This is also DNA structures. DNA. Then next part is also the fourth important part is also body nucleus. The nucleus is also prominent spherical colloidal acidophilic body. Prominent spherical means then also round shape part. It can performing the major function which can present in the nucleus, bacteria and yeast log nucleus. When the size depends on synthetic activity of cell. So sperm cells, blastomeres have smaller nucleus than oocytes, neurons, secretory cells. Secretory cells, neurons, oocytes is a large size of the nucleus. What is the position? Where is the position of the nucleus? Eccentric position. Centric and eccentric means it is also got a eccentric type of the position of the nucleus. It can associated with nuclear organization. Represents 2 degrees construction of the nuclear organization of the chromosomes, which is the nucleus nature. It can different. Then size is also different. Then location, place is also different in cell to cell or nucleus to nucleus. Then chemical composition of the nucleus is not bounded by any limiting membranes. It contains a NODNA 4 types, 4 types of the rRNAs, 70 types of the ribosomal proteins, RNA binding proteins, which is 4 types of the present particles are example nucleolin, and RNA and the RNA splicing nucleoproteins U1, U2, U3 up to U12. This is also got a nucleus important parts, important structure. This alter structure of the nucleus. This is also nucleus. Then granular components, dense fibrillar components, fibrillar center, only round in shape. Micrographically, the nucleus is also located in the peripheral or center part of the nucleoplasm. This nucleoplasm inside of the nuclear membranes. It is also fourth part of the chromatin bodies of the nucleus. Then this is also, it contains a phospholipids and orthoplasm. So this is the nucleolus, which are it contains the phospholipids, orthophosphates, and calcium ions. Also contain enzymes like acid phosphatase, nucleosides, phosphorylatase, RNA methylase. The enzymes are which present in the nucleoplasm. This nucleoplasm is important for then also in the metabolic activities of the nucleus, alter structure and function. They are a site for biogenesis of ribosomal subunit. The nucleus is majorly important for ribosomal subunits for Swedberg 40 years and 60 years. Here, three types of RNAs are transcribed: 18, 5, 5.8, 28 years. From precursors, 45 years is transcribed with undergoes processing with the help of the proteins. This is also important. Nucleolin, U3 and RNA. All the components of ribosomes are synthesized and assembled into the two types. Ribosomal subunits are transported back to the cytoplasm. Ribosomal subunits, which can, the subunits are bases, then Swedberg's 18, 5.8, 28, 40 years, 60 years. Swedberg units are, it can classify different stages of formation of ribosomes. Production, maturation are completed in three distinct regions of the nucleus. So this is a major cutter of the formation. So these are a total the nucleus structures. The nucleus structure is transcribed. The transcriptions factors and all 5S RNA and also other type of the 65S RNAs are present. This is the that is also major part of the nucleus. Then also got chromatin bodies. These chromatin bodies are important for then chromosome structures, which is also different in structures. The chromatin is also thick, elongated, or short in space, which can performing the inheritance character from generation to generation. It during the cell division, the chromatin bodies become a chromosomes. The chromosome is largest unit of inheritance. Smallest unit of inheritance is called a gene. The genes are located on the DNA. The DNA is located on the chromonema filament. This chromonema filament chromatid. Each chromatid, the two chromatids center part is called centromere. The total part of the centromere and two either side of the chromatid is called the chromosome. These chromosomes are, we will study in separate, but the chromosome or chromatid structure is not studied in clearly here. Only nucleus have four parts: nuclear membrane, nucleolus, nucleoplasm, chromatin bodies. The chromatin bodies are less, means these chromatin bodies are not covering the only single class in this type of the nucleus topic. It can separately study. That is called a chromosome. So then, what are the nucleus? How many type of the nucleus? What are the differentiation of the nucleus? First, you that this is also then the nucleus is which can gives to the two type of the nucleus structures are based on the primary nucleated cells, advanced type of the nucleated cells. the primary nucleated cell, which is called a prokaryotes. Advanced step is called eukaryotes. What are the differentiations between the prokaryotes and eukaryotes based on the character features, structure of the nucleus? This is also important. Whether this is the difference between the prokaryotic and eukaryotic nuclei. Nuclei means plural. First character, presence. In the futures, presence. The prokaryotic nucleus, not a true nucleus, called the nucleate. Basic. Nucleotic, eukaryotic nucleus, two well-defined nucleus. Well-defined means all parts of the nucleus is present. That is got defined. Second one, nuclear membrane, that prokaryotic cell absent. Without nuclear membrane, only nuclear membranes are present in double membrane in the eukaryotic nucleus. Then the third one, please unmuted. The third one, location of DNA. Location of the DNA means free in cytoplasm in nucleate. The prokaryotic enclosed inside the nuclear membranes. Inside of the nuclear membranes. Then fourth one, nucleolus. It is the nucleolus is absent in prokaryotic nucleus, present in the eukaryotic nucleus. Then also fifth one, then chromosomes. The chromosomes are which is present in the single circular DNA molecules, which are present in the prokaryotes. Only single nucleus, circular. Circular means also called plasmids and linear, the two type of the chromosomes which are present in the prokaryotes. Central part is called nucleid, then peripherally is circular, means that is called a plasmids. Circular DNA molecule, then also eukaryotic multiple linear chromosomes, line shape structures. Then 16 important histone protein. The histone proteins are absent in the prokaryotic. Eukaryotic present DNA is wrapped around the histone proteins. Then also seventh type, cell type. What are the cell type means? In the prokaryotic found in prokaryotes means bacteria and cyanobacteria. Both are important are for examples of the prokaryotic. Then also eukaryotic except bacteria and cyanobacteria remaining all type of the organism which have eukaryotic types. It is a present DNA is a then found in a eukaryotes, plants, animals, fungi. This is also then also the nuclear pores. The nuclear pores is absent in a prokaryotic, but eukaryotic is present in the nuclear membranes. It is also one important characters of the then cell division. What is the mean? It occurs by the only binary fission, but it occurs by mitosis or meiosis, means both. Mitosis and mitosis means only body cells which can double in structurally similar. In the meiosis means reparative, reducing of the chromosome number. Then double or diploid number of chromosomes are converts into the upright number of chromosomes during the formation of the reparative cells, which is also palanquins and also called a male nuclei and ovule. That is called a meiosis. Mitosis only takes place in the nucleus. That nucleus is called a eukaryotic nucleus. Difference. These are so all the differences and also completed. Now just one asking the few filling the blanks. So how to understand these topics, how to prepare the topics and how to studies and how to remembering the important points for the nucleus structures. So these are also, here is also prokaryotic versus eukaryotic, filling the blanks with the answers. The nucleus in their cells is not true nucleus and called the nucleate. And only prokaryotic cell have dark color, two type of the filling the blanks are there here. Prokaryotic cell means primary cell, only bacteria and cyanobacteria, that nucleus is called nucleate. The eukaryotic cells have a true nucleus surrounded by a nucleus. What is the true means? The nucleus which have complete all parts of the nucleus are present. That is called nuclear membrane, nucleus, nucleoplasm, chromatin bodies. In prokaryotes, the genetic material is located freely in the cytoplasm. Prokaryotes, but the dash nucleus is present in the eukaryotic cells, is absent in prokaryotic cells. So then the nucleus is only absent. That is important. The prokaryotic DNA is usually circular, where eukaryotic DNA is linear. That is also different. The 61 histoproteins are found in eukaryotes nuclei, but are absent in the prokaryotes. Then prokaryotic cells divide by binary fission, while eukaryotic cells divides mitosis or meiosis. Mitosis also meiosis. Then the nuclear pores are found in only eukaryotic cells. In prokaryotes, DNA replication and transcription occurs in the same regions. The 10th, in eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. This is also major important of the differentiations or a short questions which are in the blanks. This, all the mobile phones are in mode. So this is also differentiation. Then after this one, then what are the important of the fibular natures? Fibular center representation region of the center. Or in this is another type. Then what are the just this also make last general function of the nucleus. It is also the two sides are before adding. Then what are the function of the nuclear genes are to be produce the ribosomal RNA molecule which forms a ribosomes. Has a vital role in protein synthesis. The nucleus majorly protein synthesis. The nucleus plays a vital role in cell divisions. Cell division is important. It is the nucleus gel injured cell divisions. Then chromatin plays an important role in inheritance. The chromatin plays is important for then nucleus sap is the site of the enzyme's activity. It is supposed to participate in the formation of the spindle during cell division. The nuclear pores regular exchange of materials between the nucleus and cytoplasm. The major important functions of the nucleus. So these functions after that is also history of the nucleus is a few filling the blanks. The nucleus was first discovered the name Robert Brown in 1831. The dash suggested that the nucleus plays an important role in cell development. Matthews Sladen, Theodore Schwann. It is also one animal cell theory. The cell theory is proposed by Sladen and Schwann. That is, in 1869, Friedrich Nischer discovered the substances in the nucleus called the nuclein, now known as DNA. Nuclein, old name. DNA, old name, is also nuclein. The process of mitosis was first described by Walter Fleming in 1879. The Edward Strasberger proved that a nucleus can only arise from a pre-existing cells. It is important. The new cells are originated by pre-existing cells. Means the DNA quality comes from the old DNA to new DNA, not covering the complete new. That is, the chromosome theory in inheritance was proposed by Sutton and Bovary in 1902. The invention of electron microscopes in 1930 helped visualization of nuclear envelopes in pores. Then also the Chase experiment in 1952 confirmed DNA is the genetic material found in the nucleus. Then the nucleus is the structure inside the place close switch out moody on the phones. Then the nucleus responsible for the production of ribosomal RNA. This is also a history of the nucleus. Then filling the blanks. The nucleus is surrounded by the double membrane nuclear envelope. You know that all. The fluid inside the nucleus known as a nucleoplasm, also called a karyoplasm. Karyo means nucleus. Karyogeny, cytokinesis and karyokinesis, both are there. Cytokinesis means protoplasm division. Karyokinesis, also called a nucleus division. This karyoplasm, nucleoplasm, also called a karyoplasm. Easy. The nucleus contains a one or more dense structure called the nucleoli. Then the nucleus is often referred to as a dash of the cell, control center part, control the all metabolic activity of living cell, which is also brain of the cell. It is a fundamental part or major part of the living organism. The nuclear envelope contains a small opening, nuclear ports. DNA is the nucleus, is organized with protein into the surrounding called chromatin. Then during the cell division, chromatin condenses to form of the chromosomes. The chromosomes means it is also the chromatin bodies. Thick chromatin bodies are elongated and linear in shape to become a chromosomes. The chromosomes process starting to cell divisions. So this is the important of the RNA character and the DNA character. So, but this the nucleus is mainly involved in the synthesis of ribosomal RNA. That is nucleus. That is important. The number of nuclei in typically animal cell is usually one cell. Cells with more than one nucleus are called the multinucleated or and the syncytic or sinusoidal cells. The term karyokinesis referred to as a division of the nucleus. Karyokinesis, cytokinesis, karyokinesis, both are responsible to the cell division, two stages majorly. Then the nucleus is absent in prokaryotic cells. The nucleus controls the activities of the cell by directing synthesis of proteins. Then the double membrane of the nuclear envelope merges at the nuclear poles. Dash are the proteins around which DNA coils in the nucleus, that is called histones, which the nucleus can communicate with the cytoplasm through dash. Nuclear poles, important. Communicated means both are exchanges. The cytoplasm to nucleoplasm to cytoplasm of minerals and all components. It can exchanges, import and export. That is nuclear pores. The nucleus is not surrounded by membrane. Nucleus, the genetic material in the nucleus of eukaryotes, it is also DNA, genetic material. The process of copying DNA into RNA is called transcription. Important. Copying DNA genetic quality, one DNA to other DNA by RNA, that is called transcription. The transcription process are majorly important for parental genetic character to the daughter generation. That transcripted only RNA. But RNA is also participated in the cell division. RNA, DNA both are biomolecule in the process of the cell division. Then the term nucleus was used by scientist Robert Brown. Then the number of chromosomes human beings, you know, that somatis is 46. The chromosomes are located inside the nucleus. The nuclear envelope distinguished into the prophase of the metaphase. Metasis. The nucleus disappears during the prophase and reappears in the telophase. That is important. Prophase lobe, the nucleus also disappears. Then telophase lobe appears. It is important. Then the nucleus helps a growth, reproduction, and a metabolism. All metabolism activity. Metabolism means any type of the work, breathing, eating, also excretion, any that is also the nervous system function or hormonal function, that is called metabolism activity. The nuclear matrix provides a structural support in the nucleus. The nucleus regulates gene expression through the process of transcription and RNA processing. Transcription and RNA processing, two types, which can be then also called as the nuclear envelope, refers from the around the chromosome during the telophase. Telophase. This is also complete the all topic of the nucleus and with filling the blanks. So then now complete the today session, morning, first sessions. So please enter to the Tirupati sir. Tirupati sir.